Hello friends, welcome back to my YouTube channel Nasrin Padasalai. From today onwards, we are starting the revision series for GATE examination. If you are going to write your GATE examination under BT category or Life Sciences paper, then this video series will be definitely helpful. So daily, I will be uploading a few topics for your revision. You can use this particular topic either for studying or else in case you already prepare for GATE BT or GATE Excel, you can use these slides for revising. So first slide is regarding to restriction endonuclease or restriction enzyme. It is classified into three types, type 1, type 2 and type 3. So first property is restriction and modification. Okay. So with regarding to type 1, they have a single multifunctional enzyme engaging in both the restriction and modification. Whereas with regarding to type 2, they have a separate enzyme, separate subunit for nuclease activity and methylase activity. Okay. Next, type 3 has two separate enzymes, but both the enzymes will be sharing a common subunit. Next is with regarding to nuclease subunit structure. Type 1 has a heterotrimer structure, type 2 have a homodimer structure, type 3 have a heterodimer structure. And with regarding to cofactor requirement, type 1 alone require three cofactors. One is ATP, another one is magnesium ion and another one is S-adenosyl methionine. Next is type 2, they require Mg2 plus alone as a cofactor. Next type 3, again type 3 require Mg2 plus and SAM as a cofactor. Next, the DNA cleavage requirement varies for type 1, type 2 and type 3. First, we will be studying for type 1. The DNA cleavage requirement for type 1 is they require they have two recognition site at any orientation. That's for type 1. And regarding to type 2, they need only single recognition site. And regarding to type 3, they also require two recognition sites similar to type 1 enzyme. But these two recognition sites should be situated in head-to-head orientation. Next, with regarding to site of DNA cleavage, at which point of DNA they will be cleaving means type 1, they will be cleaving the DNA randomly approximately 100 base pair away from the recognition site. Next, with regarding to type 2, they cleave the DNA either at the recognition site or near the recognition site. Next is type 3. They will be cleaving the DNA at 24 to 26 base pair to the 3' side of the recognition site. So you have 5' side and 3' side, but this type 3 enzyme, restriction enzyme, they will be cleaving a DNA at 3' side of restriction recognition site. That is most commonly 20 to 26 base pair away from the recognition site. Next is regarding to enzymatic turnover. Type 1 alone doesn't have this enzymatic turnover property, whereas both this type 2 and type 3 will be having a enzymatic turnover property. Next is regarding to DNA translocation. Only type 1 has a possibility property of DNA translocation, whereas type 2 and type 3 they don't have any DNA translocation property. Next, site of methylation. All the three enzymes they do methylate the DNA at the recognition site itself. Next, we'll be studying the recognition sequence for some most commonly used restriction endonuclease. So first is EcoR1, which is from E. coli, and the recognition site of E. coli is GAATTC. And next is BAMH1, which is isolated from Bacillus amyloliquefaciens, and the recognition site of this BAMH1 is GGATCC. Of course, you don't need to remember all the recognition site. You can remember the recognition site which here I had given you. Only you need to remember few recognition site alone. And next is PVU2, which is isolated from Proteus vulgaris, and the recognition site of this particular thing is CAGCTG. And for HIM3, which is isolated from Haemophilus influenza, the recognition site is AAGCTT. And SAW3, which is isolated from Staphylococcus aureus, and the recognition sequence for the SAW3 is GATC. And for this ALU1, which is isolated from Orthobatus luteus, and the recognition site is AGCT. And for TAG1, which is again a restriction endonuclease, which is isolated from Thermus aquaticus, and the recognition site for this tag is TCGA. And regarding to type of N, whether the restriction enzyme after performing the activity, whether they are giving blunt end or sticky end, like that we are going to see. Only PVU2 and ALU1 and HAE, that is isolated from Haemophilus aegyptus, alone will be giving blunt end. Only three restriction enzyme after doing their activity they will be giving blunt ends, whereas rest of all enzyme, LECOR1, BAMH1, HIM3, everything they will be giving a sticky end. Okay. Next is regarding to property of DNA polymerase 1, 2, 3 from E. coli. So here we are going to study the prokaryotic DNA polymerase. So there are three DNA polymerases, DNA pol 1, DNA pol 2 and DNA pol 3 present in prokaryotes. Main function of DNA pol 1 is DNA repair and primer removal, and the main function of Pol 2 is DNA repair, and the main function of DNA polymerase 3 is DNA replication. And regarding to size of DNA Pol 1, DNA Pol 1 is 109 kilodalton and DNA Pol 2 is 120 kilodalton and DNA Pol 3 is 140 kilodalton. And regarding to structural genes present, in DNA Pol 1 enzyme there is only one structural gene is present, Pol A, and DNA Pol 2 there is one structural enzyme is present, that is Pol B, and only in this DNA Pol 3 enzyme there were five structural genes were present, that is DNA E, DNA N, DNA Q, DNA X and DNA Z. Okay, next is we are going to see 5' polymerase activity, 3' 5' endonuclease activity, 5' through 3' exonuclease activity. So what does it mean? So first we will be seeing 5' through 3' polymerase activity, which is an activity processed by DNA polymerase where the enzyme will be adding a new nucleotide at a 3' end of the strand. Next is with regarding to 5' to 3' exonuclease activity. So you should know there are two enzymes, exonuclease and endonuclease. Exonuclease will be cleaving the nucleotide at the end of polypeptide chain, whereas endonuclease will be cleaving the phosphodiester bond in the middle. So this 5' to 3' exonuclease activity will be removing the nucleotide at the 5' end of the molecule. And this activity of DNA polymerase is responsible for removal of RNA primer, which are used for the replication of the lagging strand synthesis in DNA. Okay, so 5' through 3' exonuclease activity is used for. So next activity is 3' to 5' exonuclease activity of DNA polymerase. So this is responsible for proofreading activity. So in case if there is a question asked which activity of DNA polymerase is responsible for proofreading, means you should know it is 3' to 5' exonuclease. Okay, so this activity removes the nucleotide at the 3' end of molecule, which would determine the accuracy or proofreading activity of the enzyme. So if you take 5' to 3' polymerization, it is processed by all the three DNA polymerase. Same if you take 3' to 5' exonuclease activity, which is a proofreading activity, this activity is also being processed by all the three polymerase. Whereas if you take 5' to 3' exonuclease activity, which is involved in removal of primer as well as it is involved in removal of RNA from lagging strand synthesis. Okay, so this activity is processed only by DNA Pol 1. Okay, since this DNA Pol 1 is involved in primer removal, okay, so you should know this activity is present only in DNA Pol 1 and it is not present in Pol 2, Pol 3. Next is with regarding to vectors. So vectors are classified into different types. So what is mean by vector? So vector is a DNA molecule which is used as a vehicle in order to carry few DNA segments into the host cell. So they are classified into various types and we are going to see the uses, and along with that we are going to see the maximum insert size with the example. First is plasmid. It is used for general DNA manipulation, whereas lambda insertion vector from phage, bacteriophage, is used for construction of cDNA library. Insertion lambda vector is used for cDNA library construction. Replacement lambda vector is used for genomic library construction. Next is cosmid, which is again used for construction of genomic library. Okay, so cosmid means plasmid which has cos site from bacteriophage. Okay, next is M13 phage, which is used for in vitro mutagenesis and also it is also used for DNA sequencing. Next is phagemate. It is used for in vitro mutagenesis. Along with that, it is used for general DNA manipulation like cloning and all. They will be using phagemate. Next, yak, pack, back. These are artificial chromosomes. Okay. So all the artificial chromosome, all the three artificial chromosomes are used for constructions of genomic library. Now we'll be seeing the maximum insert size in kb. The insert size of plasmid is 10 kb to 20 kb and the insert size for insertion phage is Lambda insertion phage vector is approximately 10 kilobase pair, and the insertion size for lambda replacement vector is approximately 23 kilobase pair, and the insertion size for cosmate vector is approximately 44 kilobase pair, while for M13 the insert site is very less. It is ranging from 8 kilobase pair to 9 kilobase pair, and the insertion size for phagamate is similar to that of plasmid. Next, with regarding to artificial chromosomes which are used as vector, for YAP the insertion size is 1000 kilobase pair to 2000 kilobase pair, and the insert size for YAC is very large. And okay, next for PAC the insert size is 75 kilobase pair to 95 kilobase pair. Next for BAC it is 130 kilobase pair to 150 kilobase pair. So the example of plasmid is PBR 322, PUC 18. The example of lambda insertion vector is lambda GT11. The example of lambda replacement vector is lambda ZAP and EMBL4. And the example of cosmid is PJB8. And the example of phagamate is blue script. Okay. So these are the vector. So the next video will be continuing with some other important topics. Thank you friends. Thanks for watching this video.